Hello students, welcome to the EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute, Karnal in Haryana, India. I am going to talk about the molecular database concepts from the paper Biostatistics and Bioinformatics. Molecular data refers to some known values about the molecules or some known values about their interactions with other molecules. At the basic level, the molecules are small, such as nucleotides, amino acids, sugars, lipids, vitamins, and in fact many more. These basic level molecules can combine together to give bigger, that is larger molecules called macromolecules such as DNA, RNA, proteins, hormones, vitamins, and others. Both the small and the large molecules, that is macromolecules, have associated data values about their nature, about their composition. These data values for all the molecules are compiled and stored in the databases for easy access and retrieval for use at a later stage. However, the aim of storing the data in the databases is not only to retrieve data at a later stage, but also to take out the hidden information in the data values. At the same time, when the retrieval of information is the objective, then the target is to gain knowledge, and consequently the goal is to derive wisdom. In the present module, the learning objectives are to understand nature of molecular data, molecular database concepts, data retrieval, searching databases for information retrieval, organization of the data for knowledge discovery. All molecules have their associated data values, which may be text values, such as accepted name of an enzyme, say for example 6-phosphofructokinase, a name for the third enzyme in the glycolysis pathway, or the data values may be numeric, say for example a 4-digit unique number where each digit is separated by a dot. The number may be like 2.7.1.11. This number is assigned by Enzyme Commission, and this is the standard recognized by International Union of Biochemists and Molecular Biologists, and therefore is used for storing data values regarding enzymes. This 4 digits number, which is unique for each enzyme, with each digit separated by a dot, is meaningful. For example, the first number 2 stands for transferase enzymes. The second number 7, in this example, used for all phosphotransferases. The third number is 1 in this case, and it is reserved for all phosphotransferases which transfer a phosphorus group to a substrate which has an alcohol group as acceptor of the phosphate. And finally, the fourth number, the number 11 in this example, conveys that it is the 11th enzyme in the list of all known in this group defined by first 3 numbers. In addition to this name and assigned number of an enzyme, alternative names may also be known for some enzymes, as in this case phosphofructokinase 1 and phosphohexokinase. Most importantly, the basic data about enzymes is the reaction catalyzed by each enzyme. In this example, the reaction catalyzed by 6-phosphofructokinase is the reaction of D-fructose 6-phosphate with ATP to yield 1-ADP and D-fructose 1,6-bisphosphate. Whenever this reaction is catalyzed, the enzyme involved is always 6-phosphofructokinase with assigned enzyme commission number 2.7.1.11. This reaction is catalyzed in all species, for example, E. coli, yeast, mouse, human, and so on. The reaction involved is same in all the organisms. However, the amino acid sequence of the enzyme 6-phosphofructokinase may or may not be same. In all the organisms, in addition to this data, there may be associated information which is included as attached comments to the main data. This additional information or attached comments is called annotation. In this case, the annotation is that this enzyme is not identical with enzyme 2.7.1.105, which has name 6-phosphofructo 2 kinase, that is PFK2. In the same manner, all other enzymes have data values about their accepted name, assigned number, alternative names, reaction catalyzed, and annotated information. But data values will be different for each enzyme. That is, each enzyme will have different accepted name, different assigned number, different alternative name, different reaction catalyzed, having different annotations. Let us take a look at the nature of data for some enzymes involved in glycolysis. Enzyme 2.7.1.1 is hexokinase, which has alternative names such as hexokinase type 1, hexokinase type 2, 3, 4. The hexokinase 4 is glucokinase. This catalyzes the reaction between the D-hexose and ATP to give ADP and D-hexo-6-phosphate. And the annotation includes that it can have acceptors such as ITP and DATP, donors, D-glucose, D-mannose, D-fructose, sorbitol, and D-glucosamine as acceptors. Similarly, I have already given the data about the 6-phosphofructokinase, that is the enzyme 2.7.1.11. As we look at the nature of the molecular data, we find that each of the data value is stored under a unique heading, which in database terminology we call as a field. In the present case, we have seen 5 fields with unique headings, that is assigned EC number, for example 4.2.1.11 for phosphopyruvate hydratase, which is a glycolytic enzyme with alternative name enolase. The reaction catalyzed is also shown, and this is the common field to all the enzymes. Then comment or annotation field. All these fields are common to all the enzymes. The data value for each of these fields is collectively forming one record in database terminology. Therefore, till now we have seen 4 records. In database terminology, all 4 records presented, if stored in single file, will collectively form a database. Till now we have seen records for 4 enzymes having data values for all the associated data fields. This is that format used by enzyme nomenclature database stored at Swissprot, which can be reached at XPASI server following www.xpasi.org address. This database provides 6 types of retrieval systems. First, the users can retrieve the data using the number, or they can use the enzyme class, they can describe alternative names, they can use a chemical compound name, cofactor, or search in comment lines for any keywords. Let us take an example of searching this database using enzyme commission number. Take the example of 2.7.1.1. We don't know, let us say, what is the name of this enzyme, but we know that, okay, this 2.7.1 conveys that 2 is a transferase, 7 is a phosphotransferase, and 1 is phosphotransferase having hydroxyl group as an acceptor. Now we want to see which is the first enzyme in this class of enzymes. Simply add these 4 digits in the 4 text boxes and click the search button. We find here that the data retrieved will be presented online in the browser window. Once we press the search button, the enzyme entry is displayed, and this shows that the accepted name of the enzyme is hexokinase. Hexokinase is having 4 alternative names. Hexokinase type 1, 2, 3 and 4. The type 4 is glucokinase. And the reaction catalyzes that the phosphate group is transferred from ATP to dehexose, ADP and dehexose 6-phosphate. Alternatively, let us say that we want to see the complete list of all the enzymes in this class of 2.7.1. In this case, therefore, do not enter the last digit. Enter the first 3 digits and click the search button. Once you click the search button, the complete list appears in the browser window. This lists all the enzymes which are in this class. Let us say that we want to have a look at the second enzyme 2.7.1.2, which is glucokinase. So simply click on this number so that the record of this enzyme is presented in the browser window. The data in this window shows that this glucokinase is having an alternative name, that is glucose kinase. The reaction catalyzes ATP plus D-glucose, yielding ADP plus D-glucose 6-phosphate. The reaction catalyzed is slightly different from the one catalyzed by hexokinase. Hexokinase accepts all the hexoses, whereas glucokinase is very specific to accept the glucose as one of the substrate for transfer of phosphate group from the ATP. The comment line shows that this is the group of enzymes which is found in invertebrates and microorganisms, and also lists that this is highly specific for glucose, mean this enzyme will not accept any other alternative substrate. So all these 5 fields are having associated data values. Each molecular database provides systems for retrieval of its record. The most common is retrieval of molecular data using keywords called search terms. There are 2 systems for searching databases. The first is Boolean search system and the second is concept-based search system. The Boolean search system is based on individual keywords and each keyword is separate. However, more than one keywords can be combined together using Boolean operators in Boolean search system. The second search system is based on the concept which is conveyed by the keywords. In this concept-based search system, all the words are combined to form a meaningful concept which is understood by human and is used for the retrieval of the records in the database. For Boolean search, there are 3 Boolean operators which can be used to combine keywords. Keywords are also called search terms. The Boolean operators are case sensitive, therefore use only uppercase letters. The first operator, the AND operator, directs the database to find all those records which contain all search terms combined using AND operator. This is equivalent to the intersection in Venn diagram. In some search systems, AND is the default operator and therefore when many search terms are entered in the search text box, all search terms are combined automatically using AND operator. For example, entering alcohol dehydrogenase in search text box of such search systems will be treated as alcohol and dehydrogenase, and this will retrieve all records containing both alcohol and dehydrogenase keywords. The second operator, the OR operator, directs the database to find all those records which contain either search term separately in addition to all those records which contain both the search terms. This is equivalent to union in the Venn diagram. For example, entering alcohol or dehydrogenase in the search text box will retrieve all records containing either alcohol or dehydrogenase in addition to all records containing both alcohol and dehydrogenase. The third operator, the NOT operator, directs the database to find all those records which contain the search term on the left of NOT operator and exclude all records which contain search term on the right of the operator. This is equivalent to subtraction in the Venn diagram. For example, entering alcohol not dehydrogenase in search text box will retrieve all records containing alcohol and discard all those records which contain dehydrogenase even if the search term alcohol is present in the record. Finally, in Boolean search system, phrase search is also allowed. In this case, search terms may be phrases which are having more than one keywords. To form a phrase, the keywords are enclosed between a pair of double quotes. As opposed to AND operator, the words in phrases are joined in contiguous manner. Therefore, only those records are retrieved which contain all the words in the phrase in contiguous manner. For example, phrase alcohol dehydrogenase enclosed between double quotes will behave as single search term and will retrieve only those records where both words are present contiguously. This search will not retrieve those records where alcohol and dehydrogenase keywords occur separately. However, Boolean searches does not guarantee to find all relevant records in a large unstructured database, such as bibliographic databases. With Boolean searches in these databases, keyword search often return many non-relevant records called false positives, or those records which are not relevant at all, and these are called false negatives. These false positives and false negative records are retrieved because of synonymous and polysemous meanings of the keywords. Synonymous means that more than one word or keyword in the same language have the same meaning. And polysemous means that an individual word have more than one meaning. In such cases, database searches retrieve many irrelevant, non-useful records. In addition, Boolean-based keyword search require exact typing of the spellings. If there is any mistake in typing of the spellings of the keyword, then the records which are intended by the user to be retrieved will not be retrieved. To overcome these problems, concept-based search technique has been developed. The concept-based search is very useful, especially when dealing with large unstructured textual based database such as bibliographic database containing biological literature, research. Concept search avoids the problems of false positives and false negatives. Concept search retrieves only those records which meet a particular concept conveyed by the words. Let us say that a user is interested in searching for the concept phosphorylation on serine in HPR is involved in signal transduction. Search of Google database with concept retrieves several records and presents the list to the user in an order manner with the most relevant record listed on the top of the list. In the present example, the first record in the retrieved list is stored in the PubMed Central Bibliographic Database available at National Center for Biotechnology Information. This is a research paper on the concept-based signal transduction is due to phosphorylation on serine in HPR. Interestingly, searching PubMed database at NCBI itself did not retrieve the record retrieved by Google. This shows that the concept search is implemented at Google but not implemented at NCBI. Therefore, to find the relevant records in large unstructured databases such as bibliographic databases, even PubMed, user may follow the Google route. There are three common ways to organize records in a database in a computer file. These are records in the flat file text or the records organized as table in a relational database system, and most importantly and finally, the most advanced, records stored as individual objects in an object-oriented database. To create a flat file database, one can write one record per line without writing the names of the field, and thereby avoiding the field names writing repeatedly for each record. However, to separate the data value in each field in this way, an identification mark is required to be placed between two fields. This mark or character is known as delimiter. This delimiter character may be any unusual character which is not found in the data values of the records. This may be backslash or a vertical pipe typed using backslash key with shift key on the keyboard pressed. The record for 6-phosphofructokinase is shown using vertical pipe as delimiter character to separate the data values of each field in the records. Let us enter one record using Notepad, an accessory available with Microsoft Windows operating system. While typing the record in the Notepad, the enter key is not pressed. This is equivalent to typing in a single paragraph. To write next record, however, the enter key is pressed to move to next line. In the text typing, such as with Notepad, a line continues in the same row until the key is pressed. Writing each record in a separate line is illustrated for the flat file database. The first line for first record of the phosphofructokinase has already been entered. Press enter after typing the first record to move to the next line and type the second record in this line. This is for hexokinase. Then press enter again and type the third record, then the fourth, and so on. Each record typed in this manner will appear in consecutive rows only if the first command for word wrap in format menu of Notepad is not checked. Now open the format menu and select the word wrap command. User will see that the individual records in each row are now wrapped to be displayed as separate paragraphs having more than one rows. That is, number of lines in the paragraph may be more than one, but each represents a single row for each record. Let us now store this file with a name. Storing data values for all the records in one file in this way is known as flat file system for storing database. Now we would like to search the relevant data values already stored. In text file, the data values can be searched for the desired keyword, say phosphoenol pyruvate in this case. The find command of the edit menu will highlight phosphoenol pyruvate 3 times when it is entered into the search text box. This is simple retrieval of the 2 records. The plain finding of 2 records does not reveal any additional information. It only conveys that this database is having 2 records which contain the keyword phosphoenol pyruvate. The purpose of storing all records collectively in a database is not only to store records for retrieval at a later stage, but to uncover the hidden connections among the records when the records for a particular keyword are retrieved. For example, when a user searches the enzyme database already stored in our previous slide with the keyword phosphoenol pyruvate, say in the field reaction catalyzed, 2 records will be retrieved or displayed. In this case, the data is now not isolated data values, but the data values are from a particular field, that is the reaction catalyzed. And this reveals the connection between the data values. In this way, the retrieval of data connects the 2 enzymes. The data in this case retrieved is for enolase and pyruvate. Therefore, we retrieved the reactions involving phosphoenol pyruvate and the information connecting 2 enzymes in the glycolytic pathway also. This is the purpose of organizing database with different fields on each record so as to retrieve information to gain knowledge. In this case, the phosphoenol pyruvate for the first enzyme is present in the products side, and in the case of second enzyme, it is present in the substrate side. Therefore, this connects the 2 records in the enzyme database. That is, the first enzyme produces a product which becomes the substrate for the next enzyme. So this is what we call that the 2 records, although recorded in the same database, are not able to provide data values but also to reveal information connecting the records. Another way of storing data is in the form of a table called a relational database system. Whenever ATP reacts with D-fructose 6-phosphate to yield ADP and D-fructose 1,6-bisphosphate, the enzyme involved is always 6-phosphofructokinase with enzyme commission number 2.7.1.11. This reaction may occur in E. coli, in yeast, or in human. The reaction involved is same to all organisms, that is common. But the amino acid sequence of the 6-phosphofructokinase may not be the same in all these organisms. Subsequently, when the amino acid sequence of the 6-phosphofructokinase is determined, say in E. coli, then 6-phosphofructokinase sequence may be added in the same table by adding a new field for storing this amino acid sequence data. The storage of the amino acid sequence may be in the same flat file or may be in the same relational database table. But let us say that in the future, an amino acid sequence of 6-phosphofructokinase becomes available in another species, then storing this sequence for that species in the same flat file or relational database will require for duplicating all the data values for rest of the fields. This leads to the duplicacy of the data in the database. This is known as redundancy of the data. In case we need to edit data at a later stage, will be required to edit the data in both the records because both the records have common data values for common fields, file fields, which is same in both the records. This leads to the difficulties in the data management maintenance. Relational database management system avoids the problem of redundancy by adding a new table in the same database file for storing the sequence information for each species with enzyme commission number as connection between the two tables, that is the main table and the sequence table. Whenever a user searches the database for the sequences of the enzyme 6-phosphofructokinase, then the management system of the database will first determine the EC number of this enzyme from the main table, which is 2.7.1.11, and then use this EC number to search the second table, that is sequence table, to retrieve all the sequence records for this number. In this way, the problem of redundancy is avoided by storing the data in a separate table in the relational database system. Further, when the sequences of some other enzymes, other enzyme in other species are also determined, then the same may also be included in the same table. In this way, same database having a separate table for sequence data continues to grow without duplicating previous added data about the reaction catalyzed, about its name, about its annotation, and so on. Relational database management system has additional advantage of integrating databases. The sequence information may be stored even in a separate database. In fact, UniProt KB database houses the amino acid sequences of all the proteins, including the enzymes, in one separate database, that is the sequence database known as UniProt KB. When the user searches the sequence database for 6-phosphofructokinase using either the enzyme commission number or its name, this database will provide the sequences of all the enzymes in the sequence database. In this way, the database available for enzyme, the databases available for sequences may be integrated using relational database management system. The development of relational database management system helped in retrieving not only the data but information also. However, the development of object-oriented databases helped further by adding the property of knowledge discovery. The classical experiment of Anfinsen with RNAs revealed that the information about folding of the enzymes is encoded in their amino acid sequence. Since an enzyme is an object and knows how to fold into three-dimensional structure, then this information may be used by the database programs to compute the three-dimensional structure by taking the information from the sequence. In the object-oriented database, the individual objects, for example, knows how to fold into three-dimensional structure to catalyze that reaction. Therefore, when the object knows how folding can be achieved in 3 dimensions, then if a program can be developed to use this sequence information to fold in 3 dimensions, there will be no need to store the structured data for 3 dimensions in a separate database. This information of folding enzyme into 3 dimensions is encoded in the sequence, and the same may be used to fold enzyme in silico by using computer programs. But this is a future expectation when scientists will completely understand the folding rules. However, this form of storing data in the form of object-oriented databases will ultimately be useful for knowledge discovery. In bioinformatics, simulations for a given object like an enzyme under the changing environment of pH, temperature, metabolites will enable the elucidation of regulation of biological processes such as biochemical pathways or signaling cascades. Dear students, let us now summarize what we have learned in this module so far. So far, we have learned about the nature of molecular data. We have seen that all the molecules have with them associated data values such as their name, their molecular weight, their sequence, and many more data values. These data values show us that the nature of molecular data is very complex, and all this data about the molecules is stored in the molecular databases. And therefore, we have learned what molecular databases are, what are the concepts, what type of molecular databases are available. In addition to the molecular databases available, we have also learned about how to retrieve the data from the databases. We have seen that there are two systems in searching the databases. The first is Boolean search system, which is based on keywords, individual keywords, which can be combined together. And we have seen the second search system, which is based on the concept. And this concept based data retrieval is actually much more useful for searching databases. And when we search the databases, we not only target to retrieve the records, but our intention is when the records are presented in the list, the connection among the records are also displayed. This is what we call information. This information retrieval is the target when we actually store data in the databases. Otherwise, in a list, if the all records are separated from each other, then this is not meaningful from retrieval of the information point of view. That's why very complex database search systems as well as the advanced database storage system such as object-oriented databases has been developed. The organization of data for storing the data in the database is actually with the final or the goal of the knowledge discovery. We are interested in discovering the knowledge from the data through information so as to ultimately derive wisdom which can be used for better human and other living systems, health, nutrition, and environment. Dear students, I thank you for visiting EPG Pathshala.